Today we discuss different types of microscopes so we can understand what type of technology we use to observe cells, and our success criteria is being able to differentiate between the light, electron, and scanning microscopes. So the microscope you are most familiar with is probably the compound light microscope. These are the ones that we'll often use in classrooms where we're basically backlighting a very thinly sliced sample so that we can see through it. And these were, in fact, the first types of microscopes to be invented. They are fairly simple compared to the other ones, but they have very good resolving power, at least when we want to look at the scale of cells themselves. But there are other types of microscopes too. We can also have electron microscopes. Big difference between a light electroscope or microscope and an electron microscope is that the electron microscopes, instead of using light, they're actually transmitting electrons through a specimen. Or if it's a scanning electron microscope, they're actually sweeping electrons over and using the information about it to produce a three-dimensional image. So images like what we see in the bottom right, which is a whole bunch of white blood cells, no one is taking a photograph of images like that. It's actually an image produced by a scanning electron microscope. And these electron microscopes look a bit different than what you might think of as a microscope. They look more like some sort of chamber that you should be doing nuclear physics in. But this is a microscope from the University of Alberta. And what they're doing is they're using electrons to scan an image of a cell at very high resolution. There are other types of scanning microscopes as well. You can use a scanning laser microscope. Instead of using electrons, these use a laser, and what they basically do is the laser beam is directed at a sample at different angles, and it's basically taking a whole bunch of two-dimensional images and compiling them together to make a composite that shows three-dimensional depth. And we use laser scanning microscopes on samples that are too thick for a compound light microscope, but also don't need the same level of resolution as in detail as an electron microscope. And we also have something known as a scanning tunneling microscope. This has much more resolving power than an electron microscope does, so we can use it actually to get images of molecules like DNA. And sometimes chemists will use this as well in order to try and guess at the structure of an unknown molecule. And these ones do use electrons too, but what they're doing is they're actually using an electrical probe and measuring how easily the electrons are flowing between the probe and whatever specimen they're studying. And that information can then get relayed back into a computer, and the computer can use that to approximate what the shape of the molecule will be.